Hello and welcome everyone. In this podcast we're going to continue our discussion of macromolecules. In previous podcasts we have talked about the first two macromolecules. We talked about carbohydrates and proteins. What we need to still talk about are nucleic acids and lipids. We're going to talk next about nucleic acids. Let's first begin by talking about the functions. Nucleic acids. The first function we can think of is that they store information. The second function related to storing information is that as they store this information, nucleic acids are also the unit of heredity. On the previous podcast we talked about the central dogma. Remember that was going from DNA to RNA to proteins. DNA and RNA are nucleic acids. And so you can see how DNA and RNA has this third function, which is they code for proteins. Now, in some cases, some nucleic acids have a direct function. And that is relegated to RNA molecules. And not all RNA molecules, some special RNA molecules, which have very important functions. Okay, these are the four main functions I want you to know about nucleic acids. Next, I want to tell you the types of nucleic acids. And I've already mentioned them, but let's just write them down here to be complete. The first include DNA, and the second include RNA. Now, there are many types of RNA. You've probably heard of ribosomal RNA, transfer RNA, and messenger RNAs. But we now know there are many other kinds of RNA as well, which we probably won't talk about in this class, but when you get to genetics and cell biology, we will talk more about RNA molecules. Okay, I'm going to erase this now. So, as a repeating theme here, I want to talk about these monomers and how they're held together to form polymers. Just like with carbohydrates and proteins before, nucleic acids are a repeating string of monomers. And this repeating string, or rather I should say the monomers, are nucleotides. And the bond that holds them together is called a phosphodiester bond. You should know those two things. Now, you're not going to have to draw a nucleotide, but you should know that nucleotides are made up of three things: a sugar, a nitrogenous base, and a phosphate group. These three things are important for a nucleotide. Again, you're not going to have to draw this on an exam, but I just want to show you what it looks like. This is your sugar, and it can be either ribose or deoxyribose, depending on if it's RNA or DNA, but we'll come back to that later in the semester. You have a phosphate group here, and over here you have your nitrogenous base. When nucleotides come together, they come together at this OH group on this sugar. So the next nucleotide is going to be below here. And this is where that phosphodiester bond forms to help make this linear sequence of nucleotides. Now remember, we said that in proteins they have these amino acids that are characterized by an R group, or differ by an R group. Now nucleic acids are similar in that way. And they don't have an R group, but these bases can be one of four bases. They can be adenine, which we often just call A. They can be thymine, which we often just call T. They can be guanine, which we often just symbolize as G. They could be cytosine, which we often just call C. And they could be uracil, which we often just call a U. But based upon if you have A, T, U, or G's or C's, that ultimately will tell us which protein will be coded for. Okay, now that we've drawn the basic structure of what a nucleotide is going to look like after it begins to form, I'm going to erase this and talk about how the overall nucleic acid looks. Particularly, I'm going to focus on DNA and looking at the double helix. On this side here, I'm going to draw kind of a generic looking nucleic acid. I'm going to draw the sugar here, the phosphate here, and the base I'm going to put here. I'm not going to put B, I'm going to put a specific base here in a moment. And then this sugar connects with a phosphate to the sugar, and then to the next base, and then it connects to the phosphate of the next nucleotide. And we'll just draw one more. Now remember, this is one nucleotide. I'm going to put just NT for nucleotide. This is the next nucleotide, the third nucleotide, and then our last nucleotide here. Nucleotide includes the phosphate, the sugar, and the base. I'm going to draw in the nucleotides here, just the letter. So guanine, cytosine, thymine, and adenine. Alright, I'm going to draw the other strand of the nucleic acid to help construct the double helix. I'm going to draw the bases first. We'll learn more when we get into genetics, but the second strand here goes backwards. You don't need to know why that's the case right now. But it's going to have a sugar with a phosphate, just like we see over here. Then it's going to connect up here to the next phosphate with the sugar connecting to the base. The phosphate, sugar connecting to the base, to the phosphate, to the sugar connecting to the base. Now, I'm not going to make you draw something like this on the exam, maybe in the activity, but not on the exam, but you should recognize it. I might give you something that looks like this. And then I'm going to ask you, perhaps, to fill in the bases missing. Which bases should be there? And to be able to do that, you have to know these rules. They're called Chargaff rules. I'll write it down here. I can't imagine me asking you who Chargaff is, but just in case you're curious, he was the one that came up with these rules. We know that G's bind to C's, and C's bind to G's. And we also know that T's, thymines, bind to adenine. In addition, we know that there are 3 hydrogen bonds between the guanines and the cytosines, and 2 between the thymines and the adenines. So if you had to complete this nucleic acid structure here, you would add a C here, cytosine, because it binds with G, G, because it binds with C, A, adenine, binds with T, thymine, and then thymine binds with adenine. You should finish this off by including the base, the hydrogen bonds between them. There are 3 between the G's and the C's, and 2 between the T's and the A's. Like that. Some ways to help you remember a couple of these rules. G's and C's, they bind with each other, and I've always remembered this because both G's and C's have like this curved nature to their letter, to their capital letter. And T's and A's bind with each other, and T's and A's you could write with just straight lines. So they share those kind of characteristics. If you're having a hard time remembering that G's and C's form 3 hydrogen bonds between them, whereas T's and A's form 2 hydrogen bonds, the way I remember that is both G's and C's rhyme with 3. T and A, both of those do not rhyme with 3. T does, but not A. So you have to first remember G's and C's bind together. That they both rhyme with 3, so you have 3 hydrogen bonds between them. And just, I should have mentioned this before just to remind you. We did on the other side, so let's do it here. So that's a nucleotide. Sugar, phosphate, and the cytosine. Sugar, phosphate, base. Sugar, phosphate, base, and then sugar, phosphate, base. Another just side note, remember right here, these are our phosphodiester bonds. I'm just going to make a little line by here everywhere there's a phosphodiester bond. It's always between the sugar and the adjacent base phosphate of the next nucleotide. So here I'm just going to draw a black line and say phosphodiester bond. The last thing I want to do is erase part of this slide here. Now I want to remind you that the DNA structure itself doesn't look exactly like this. It twists and folds in a very unique way, and we're just going to draw it like so, as a double helix. RNA is also nucleic acid, and RNA does not have this double helical nature to it. It's a single strand of nucleotides. We'll talk later as the semester goes forward more of the differences between DNA and RNA. But for right now, this is all you need to know about nucleic acids. The last thing I want to talk with you about is the 4th macromolecule. And if you've been keeping score at home, that's last macromolecule includes the lipids. As we've done in the past, let's first start talking about some of the functions of lipids. They're a great source of energy storage. Lipids themselves can be hormones, so they're important in producing hormone production. And they're incredibly important in holding together this cell membrane. Now, one common characteristic of all lipids is that they are all hydrophobic. You've probably seen an example of this if you've ever had, say, a cup, and you mix in it water and oil. Soon what you'll discover after you let it settle is that the oil comes to the top, whereas the water is on the bottom. Because the two do not mix. They do not form a solution with each other. So they separate. And that's because lipids, including fats, are hydrophobic. Okay, let's erase this. There are 3 kinds of lipids we want to talk about. They include fats, and sometimes we might call these fatty acids. It would include phospholipids in the next type of lipids. And the third one would be steroids. Let's first talk about our fatty acids. So, fatty acids. And sometimes we just call these fats. A fatty acid includes 2 parts. A glycerol molecule plus 3 fatty acids. So a glycerol molecule we could draw like this, with 3 carbons. And we're going to have hydrogens off of here. The carbons are connected. And then, so this here is our glycerol molecule. Next, let's draw the fatty acid portion of it. The fatty acid portion comes off like this. For this first one, I'm going to draw the carbons. I'll go ahead and draw it for all of them. Off of each of these carbons, there is a hydrogen. Rather, there are 2 hydrogens. I'm not going to have the space to draw these on all of them. I'm going to draw it on the top one here. But you should know that there's 2 hydrogens off of each of these carbons as I have them drawn. There will be an exception that I'll talk about in just a moment. And this last one has 3. Now all of these would have the same kind of hydrogens written across it. Now this particular fatty acid we would call a triacylglycerol. So I'm going to write that here. Triacylglycerol. I should spell tri correct, because it has 3 fatty acids and a glycerol molecule. I'm going to erase the top part of this whiteboard because I want to use the picture below to talk about the difference between a saturated and an unsaturated fatty acid. As I have this written now, and assuming I have hydrogens off of all of these carbons here, we would call this a saturated fatty acid, or we could call it saturated fat. And we say it's saturated because each of these carbons contains all the hydrogens it possibly can. Now if you remember, some characteristics of saturated fats is that they pack tightly together because of this nice linear array of the carbons. Because they can stack nicely together, saturated fats are solid at room temperature. They are also less healthy for you. A diet high in saturated fats is a risk factor for heart disease. Now let's use this same diagram to talk about unsaturated fats. As we said before, saturated fats are saturated because the carbons are saturated with hydrogens. So let's, on the bottom strand here, talk about how we can make this saturated fat into an unsaturated fat. And essentially we do that by making at least one double bond here. We could have 2 double bonds, but when that happens, we reduce carbon's ability to bind another hydrogen. So in this case, this carbon will be bound by 2 hydrogens. This one will only be bound by 1, as this one will also only be bound by 1. This will be bound by 1, as will this one. This one will be bound by 2, and 2, 2, 2, and 3. Why does this matter? Well, it's maybe a little hard to see here, so what I'm going to do is draw it up here, but more in a kind of a line manner. So you have your glycerol molecule still. The first 2 are like this, very ordered. But however, this one, because of those double bonds here, what happens is, but this one, because of these double bonds, will now fold a little differently. And it'll be kinked like that, so to speak. And because of this small kink in the carbon chain here, they don't stack up nicely with each other. And so they're much more unorganized. And because of that, unsaturated fats are liquid. Saturated fats are solid because it's a nice stacking of the fatty acid side chains. In this case, they can't stack nicely with each other, and because of that, they're liquids. These are more healthy for you. I've drawn 3 black arrows here to signify the characteristic bond of fats. And these are, I'm going to draw it over here where I have a little more room, called ester bonds. E-S-T-E-R. I have said before that polymers are the result of linking similar or identical monomers to each other. I have also said that in the earlier podcast that of the macromolecules, lipids are the one that is not a polymer. Carbohydrates, proteins, and nucleic acids are all polymers because they have a repeating monomer subunit. For a few reasons, lipids don't follow this rule. For one, as you look here, we don't see a common repeating unit because we have a glycerol molecule here holding these 3 fatty acids together. Another reason we can think of lipids as not necessarily being polymers is that lipids are not always the same kind of structure that we see here. I'm going to erase this real quick because I want to talk about the other two kinds of lipids. Remember, one of these other kinds were the phospholipids. The other one are steroids, and we'll talk about those in a moment. The thing about phospholipids that's important to note is that they don't have 3 fatty acid tails. They have a phosphate group here, and then they have 2 hydrophilic tails that are made up of carbon chains, much in the same way that the fatty acids in fats are. So 2 things I want to make sure are clear here is that these are hydrophobic lipid tails, we call them. The phosphate group here, I didn't mention it when I said it the first time, but they're hydrophilic. The nature of the phospholipids make them suitable to form membranes, and we'll talk a lot about membranes in the future. But for now, what you should know is that a membrane consists of these phospholipids, like so, with the hydrophilic phosphate group on the outside and the hydrophobic tails on the inside. And we'll come back to this later. Just to kind of preview what's to come. The third kind of lipids are steroids. I'm not going to spend a lot of time talking about steroids, but you should know that steroids are typically structures of fused rings. I'm just going to draw them as squares, but they're not usually squares. They're usually different kinds of shapes. And they can have additional carbons coming off them, like so. And these are important in regulating various functions in our body. One that you've probably heard of is called cholesterol. Cholesterol is a steroid. And it is a lipid. Even though we don't necessarily think of them as lipids, they really are lipids. And they're lipids because they share that common characteristic in that they are hydrophobic. Alright, that's all I have to say about lipids and nucleic acids for this podcast. If you have any questions at all, please make sure you let me know. If not, I'll see you in class. Bye.